So now that we know the basics of NGS, now how to analyze what situation we are going to analyze once we have done with that. Now, why in this particular topic, I'm going to talk about breast cancer. I have a very good success stories with breast cancer also. So now in breast cancer, how are we going to use NGS? We'll see. Okay. So this is the approximation that global estimate incidence of breast cancer in females in 2018. They have given this report. So breast cancer, as we know, is now a very, very common type of cancer among women. Okay, and it is also very lethal. Okay, so once the patient is being diagnosed with breast cancer, once they see a lump or see a tumor, first thing they will do is a biopsy. Okay, once the biopsy is done, we are going to do something called as IHC. IHC is not any very advanced molecular technique. It's called immunohistochemistry. Okay, there are certain markers that they will put on the cells. Okay, so now what happens? If there is a lump, they will do a FNAC, or they will take an excision of that tissue and they will fix it. As I told, they will fix. So that's called FFPE. In paraffin, they are going to fix it, embedded tissue. So that tissue they will take and they will make slides of it. On that, they will do IHC testing. On that, what they are going to do is they are going to add some antibodies to detect. First marker is HER2. Okay. HER2 marker as per CAP and ASCO guidelines. So these guidelines suggest that first thing they have to do is they have to do a HER2. HER2 is a receptor that is present on the cell. Okay. In case that is present, very good. They will stop there itself diagnosis and they say it is HER2 positive breast cancer. They will start on some hormonal therapy like Herceptin. They will give and there only they will start. Okay. In case HER2 comes negative, next they have to do is ER and PR. Those are also estrogen and progesterone. Those receptors also they will check. Nowadays, they are doing all three together, ER, PR, and HER2 together. But ideally, it should be in a sequential way. In case ER, PR also comes negative, next is what they go for. They classify that type of cancer as TNBC, triple negative breast cancer. Triple negative is HER2, ER, and PR. Once these three comes negative, there are chances of BRCA mutation, BRCA gene mutation more in them. That is when they go for this TNB. So BRCA testing. So when does this BRCA testing? The advice is when the patient is classified as TNBC, mostly when they are male patients. Nowadays, male breast cancer, I think I don't know, when I saw first time that male breast cancer had come for my analysis, I was like, what, male cancer? Male breast cancer? Yes, male breast cancer is also equally there in the population. Then, is there any family history or not? And then, if the patient are going to be considered for any olaparib, like any inhibitor type of therapy or not. Based on that, those people will be given for NGS BRCA testing. Okay. So, apart from BRCA, I was telling, these are the other genes that are equal, like we can see their frequency in breast cancer, mutations in these genes. Okay. But if you can see, BRCA1 and BRCA2 are highly mutated. Okay. We have seen mostly nonsense variants. Okay. So now what is a nonsense variant? A variant, a substitution that is going to induce a stop codon. Okay. Once the stop codon comes, abruptly the transcription or translation will stop. And a protein that is formed from there is going to be a very truncated protein and it is not going to do its job. Okay. So now why we are going to be interested more with BRCA1 and BRCA2 in breast cancer patient is these are called DNA repair genes. Okay. So whenever there is going to be a problem, yes, our body is continuously undergoing lot of divisions, mitosis, meiosis, replication. So many things are undergoing in our body continuously. So then at that point, there are high chances that there will be error. Okay. So to proofread those error, these genes play a major role. These are called DNA repair mechanism genes. When these genes itself are going to become a problem, that's when the cell will not be able to combat those issues and they will go into a cancerous state. Okay. So cancer cells are nothing but our normal cells that have started to act differently. Now, why they are started to act differently is what we are going to see through NGS. Okay. Which gene has made it? Okay. Especially in breast cancer, when they have been classified as TNBC, we go ahead and do BRCA1, BRCA2 testing for them. So there are two ways of doing BRCA1, BRCA2 testing. One is by NGS. Okay. NGS is what we have seen fully, right? So with that we can do. So that will tell you any point mutations, any small deletions, small insertions, small indels. Okay. It can tell. Okay. However, NGS is not very specific towards large deletions. Say large deletions like exon deletions and all. It may not be able to tell with full confidence. So that time we have another way of testing that is called MLPA. MLPA is also, it is highly patented. Unlike NGS, MLPA is highly patented, and that patented is by Holland, MRC Holland. They say only they have the primers. If any lab, and it is based on Sanger technique. So the primers you will definitely get only from there. However, NGS are little open source, like you can design your, sometimes you can design your own primers, and there are a lot of companies that will help you design primers suitable for either Illumina, Iron Torrent. Iron Torrent is also a little close, but Illumina, yes, there are so many companies that will help you design your own primers. Okay, so BRCA, either you can do by NGS. NGS is the first thing they will do. Second thing, in case NGS comes negative, especially when it is a TNBC and germline, in case it comes negative, then they will go for MLPA. MLPA will detect large deletions in the BRCA1, BRCA2 gene. And BRCA1, BRCA2 are very huge genes. BRCA1 has approximately 21 exons, I think, and BRCA2 has up to 24 exons till now. That is what they have seen. So these are huge genes, okay? So now why should we do NGS for breast cancer patient is, we need to understand, okay? When there is a particular type of gene that is mutated, then we will know what type of treatment we should do. So this picture explains very, easily why we should do an NGS is, in a population, if this entire population we are going to give, for this entire population we're going to give the same type of drug, maybe a few won't respond only. That's because they harbor a particular type of mutant, a particular type of variation, right? So when we do NGS and then categorize these patients as, say, BRCA1 positive patient, BRCA2 positive patients, no BRCA variants, PIK3CA mutation, or say some other type of, say, PAL-B2 is also there. So if these mutations are going to be there, then to treat them separately is very, and they will also respond, okay? So your diagnosis will be easier, faster, and treatment procedure will be very accurate. So for these reasons, we have to take these patients for NGS. Yeah. So now in BRCA, though, I'm going to more say about BRCA gene variants, because that's, as I told, they account for the largest population of variants. So if you see, BRCA mutations are the most common genetic variants in the breast cancer, and especially when HER2 is negative, up to 10%, and TNBC, if you see, they are up to 10 to 24%. EBC is nothing but early breast cancer. Okay. So however, this prevalence differs from ethnicity to ethnicity, race to race, and age to age. Age to age is very important, because earlier it was told that TNBC less than 50 should be going for germline TNBCs, but now it is not like that. It is changed. Okay. So now why we have to see for BRCA is, as I told you, BRCA is a DNA repair mechanism gene. Now what this gene will do is, it will go and wherever there is damage in the gene, it will go and help the BRCA. But now when BRCA is not there, there is one more person called PARP who will come and try and help the situation, but he may not help it correctly. So we have PARP inhibitors. So if this PARP inhibitor will come and inhibit this, what will happen? It will lead the cell into death phase. In case it does, in case PARP does not do the proper damage therapy, and if it goes into cell progression, it will convert into cancerous cells. So this, in case we inhibit this PARP, the DNA collapses and will go into apoptosis, which means the number of cancer cells is going to reduce directly. Okay. However, cytotoxicity is there. Okay. So I will go through a case study, a very interesting case study, but I am going to use a commercial software. So my software is from Thermo Fisher company, and this is called Oncomine or OKR, they say, Oncomine Knowledge Reporter software. So this does only oncology reporting. However, Thermo Fisher also has their own reporter software called as Ion Torrent Reporter software. Okay. So this Oncomine Reporter software is based on Oncomine database. So there is a database, huge database. Okay. The database has huge data through which they have developed this Oncomine Reporter software. Okay. This is the reporter software. So this is the reporter software. Okay. So in case we are doing for the first time, we'll have to create the analysis. So which means those BAM files, your VCF files, your all those files you remember, from there you will have to insert into this. Okay. Now I have some already analyzed some data. Okay, great. So now if you see, these are the three genes, that is three variants that is present in this person. Okay. So first one is the first one is this BRCA1 Q210, and this means termination variant. Okay. So in 210, it is undergoing a stop loss has happened. So this is classified as loss of function. That means there is a truncated protein. This variant class is called truncating, and it is a nonsense variant, and the allele frequency is 2.70. Now, what is this allele frequency? I will tell you is, say now, after the sequencing is happened, we have huge reads, right? So those reads will be aligned, and in how many of these reads this variant is seen is the frequency. Okay. 2.70 is too less to be considered. Okay. However, the ClinVar suggests it as pathogenic variant. Okay. So now second variant I will go. This variant can be reported. It has a better frequency of 4.63. Okay. So this is again a termination variant. It is in BRCA2 gene. This also leads to a loss of function, truncating, and is pathogenic. Okay. So we will see some other patient. Okay. So this patient does not have BRCA1, BRCA2. She has some other variants, but you can see here it is benign. This one is a deletion variant. However, 1.93 is not a good allele frequency to be reported, but this is a deletion variant. Okay. Okay, so now if you see, this patient has 18 variants. She has how many variants? 18 variants. So the first variant is in a gene called PIK3CA. What is happening? H1047R means the histidine in this position is changed to arginine. So it is a pathogenic variant. And what is the percentage? 79%. So my sample has passed the QC. I have aligned it and my uniformity is good. My on target is good. Now my allele frequency is good. And this is a pathogenic variant. So I will report this variant in her report and suggest that she is a PIK3CA variant BRCA patient or breast cancer, not BRCA patient, breast cancer patient. Okay. So these are the other variants. Okay. I will show you how it is. BRAF is another variant they have seen, but it is only very less. So we can't report this. So KRAS, very less variant, very less frequency again. Again, very less frequency. PTPN is also a pathogenic variant. However, very less frequency. You can see it is a gain of function. So this may have induced the cancer, but the frequency is too less. We cannot consider it. Okay, TP53. See, conflicting interpretation of pathogenicity has come. Now, when these type of variants come, we have to use our gene interpretation tool. We have to read about this variant in the databases. We can use ClinVar Miner. We can use ClinVar and study about this variant and then put those categories in our variant interpretation tool and understand if this variant is going to be pathogenic or not. Okay, so like that, every variant he will have to be seeing. Okay. So this patient does not have a BRCA variant. She has a PIK3CA variant. So this is another set. It is after BRCA 1 and 2 in South Asian population. PIK3CA is the next leading mutated gene in the breast cancer patients. Okay. As I told you, I showed you what is called the Oncomine software. So this is a commercial software. It is designed by Thermo Fisher. They also have an ion reporter software that comes with the machine. So that this is a commercial means we'll have to be paying per report charges. Whereas this ion reporter software is unlike that. Oncomine can only do cancer related analysis, whereas ion reporter has other say infectious diseases, microbial, and they have inherited diseases also. Okay. And then this software is designed and it's because it is coming from a database called Oncomine and they have only Oncomine related data. Okay. So now once that is done, this is another very important validation for us is integrative genomic viewer. So this is called IGV. So once we have said this, yes, I have read about the variant and it is correlating with the disease condition. Now I have to see, I have to check. I told you the alleles, right? So those allele percentage is coming from this. So the red and the blue ones you can see, the red is the reds are the forward and the blue are the reverse reverse reads of our DNA. Okay. So you can see here, this is a heterozygous type of variant, which means it is there in 50, say more than 50%. Okay. So you can see here, if it is going to be homozygous, then all my reads will have this variant. So this is a good, comparatively good IGV because you can see most of the reads are there and this variant is there in my at least 50% of my variant. Okay. IGV also you can download from online. It is a standalone tool. You can just put IGV integrative genomic viewer. You can download this and you can upload your chromosome. Like you, once you have done all this alignment, all that, you can upload the VCF file. You can file into it, and you have to give the genomic position, chromosome positions in that. Okay, once you upload, you put that, it will tell you like this. It will show a very clear image in that position what has happened. It is highly position specific, and you can also do comparative analysis in this, like by adding more than one sample to it. Say in case you're doing 5 samples, and in all 5 samples you have got the same variant, then it can be a machine induced error. So to know that, if you add all those 5, and if you see those all 5 are there, then you have to do a deep genotype phenotype correlation, and then we have to do a validation. Validation is, you can also do a Sanger validation once the NGS picks up a particular variant. Okay. So then with this, the other challenges, okay, what are all the challenges that is there in NGS is, okay, one is technology wise, it's very new. Okay, and there are very few standard protocols that has been validated till now. Okay, and all new NGS workflows, say in case you are going to design any panel, it requires a huge amount of time, expense in validating it. Okay, now, and you need specialized machines for it. And of course, as I told you, you need extreme bioinformatics skills to understand this, and clinical interpretation of basis plays a major role. And one more biggest challenge this NGS is facing is tumor heterogeneity. So what is tumor heterogeneity is, in case of cancer cells, what happens, not all cancer cells are going to act in the same way. They are going to be, those cells, because they are not as per the nature, every cancer cell will be may be different. Okay, so that will increase the tumor heterogeneity. Once the tumor heterogeneity increases, what happens, as I told you, that allele frequency will come down, my calling rate will come down, then the tumor percentage, if it is down, if it is less, then doing NGS is difficult. All these issues are there. Okay, so now how are we going to find solutions? So we have to properly understand how we have to validate our variant. So what are all the tools are available? In case we are not satisfied by one tool, we can use multiple tools or multiple bioinformatics tools to understand. And there are also certain bioinformatics tools that will predict the pathogenicity. Say we have SIFT, we have Polyphen, and then we have mutational detect. So all these tools will tell that if in particular point there is a mutation, will it cause a disease condition or not. So those on in silico tools we can use. And then to reduce the manual errors, more of automated machines are required. So these are some ways through which we can combat the current disadvantages. However, a more interesting technique is being designed, and it's evolving, it's still evolving, is detecting mutations in CTCs and cfDNA. So now what is CTC? Is circulating tumor cells, and cfDNA is cell-free DNA. Okay. In the beginning of this session, I told they can use blood cells in 2 ways. Okay. One is taking the genomic DNA. Okay. The second one is we are going to take the DNA from the plasma. So in the plasma, so now what is this? Is a very tricky thing, right? I'll tell you. Now what happens now when a cancer is formed? Okay, so when cancer is growing, the cells, as I told you, it is not as per the nature. They may have the capability of dangling. Okay, so they will dangle and they will move out of the primary source and they will come into the bloodstream. Or sometimes, since they have not properly formed, that cell will burst and they will put all those DNA into the bloodstream. So those DNAs are those DNA or those genomic material that has come out from the cell is called cell-free DNA. And these cells that has come out from the primary origin are called CTC, circulating tumor cells. So the CTCs when you see more CTC is when there is a metastatic condition. That is when there is a primary and the cancer is already spread to the body. That is when we see the CTCs in the patient. However, taking the CTC or cfDNA is highly tricky because their concentration is very, 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 very low. It requires huge amount of expertise in extraction procedure, in purification procedure, and in analysis procedure. Okay, to combat this, okay, this is the fourth generation. This is the very, very interesting, and it fascinates me so much because for 6 years I've been in NGS. I have seen lot of understanding if my variant is really accounting to a problem or not. Because of major reason is tumor heterogeneity. I told, I used to feel like, will this be a driver mutation or not? Will this mutation cause the disease or not? Should I give this in the report or not? Will it change the treatment strategy? If it change the treatment strategy, will it help the patient or not? In the 6 years of my career, I have faced this a lot of times. Okay, but this fourth generation, or you say single cell genome sequencing, is an excellent strategy. However, this very, very, till now, since we are learning, we are growing, like how from Sanger to NGS we have come, this will also or may also take time. But now there are many companies that are pioneering this research, this place, this they are trying to find out, combat the small disadvantages. And what happens here is they take only a particular type of cell. Okay, only one particular type of cell is being taken by either by laser capture method or by fax. Fax is nothing but flow cytometry, that's all. So flow cytometry will sort it. Flow cytometry is nothing but you will put the cells in the machine. It will sort it based on the markers. Okay, so that is fax. And then some microfluids, like say some beads. With those beads, you can take out only a particular type of cell. Only one cell you will take it, and you're going to take the DNA from that. Some people say that we can culture those cells and then take the DNA. Some people do multiple displacement amplification. So with that also we can, so this is the MDA technique. So that is also giving them results. Then they will make a library, then they will sequence it. So now whatever the data is coming in this sequence will be of that one particular type of cell that we have chosen. Right? So with that, what happens is our confidence of telling a variant is high. So we are going to choose this cell also in a very tricky way, that it is definitely a cancer cell. Okay, it is definitely a causative cell. Okay, that way we are going to take it. And then if we do it, identifying and analyzing and reporting will be more confident. Like with more confidence, we can report and tell the patient, yes, this is the driver mutation we are facing. Okay. So that was one. And this is another, another visualization which I was very, very, excited about when I saw this first time is, say, I told FFPE block, right? In that tumor heterogeneity is there. Say this is that tumor, or this is that tissue. Okay. So this blue, yellow, green, all that you can see, those are the different cells. So in this blue cell is of one type, this yellow cell is of one type, the green is of one type. Okay. So this is another technology. I would like to show an interesting video. I think it will be definitely interesting. I would also say people to please pursue towards this research. It is more important for us. This is called a Visim spatial gene expression. Please listen to this video with me. It's a wonderful 2-minute video. And this is designed by 10x Genomics. Is it audible?
Yes, ma'am, it is audible.
Thank you. Thank you for watching. So this technique, what it does is, it is going to take RNA or the DNA. So this is also called meta-transcriptomic analysis or data analysis. That what is going to take is, it is going to, in real, it is at that point of time, whatever RNA was there, from every cell, it is going to take out and put it in a particular well, in a single well, and then it is going to amplify, and then it is going to tell what are all the RNA or the DNA is being basically RNA is expressed at that point of time. So what it does is, it helps what it not only helps the doctors to decide what type of treatment it should give, but also till what time it will be helpful. Will that cell or will that cancer take up any acquired mutations that will cause resistance or not? So that level of technology we are at, and this is it's a very growing technology. If any of you get opportunities to read about this, or to learn, or to undergo any training on this, I would highly recommend, because this is the future. This is the future of cancer diagnosis and treatment. This will help us real time how we are going to detect, diagnose, and treat the patients. So this a small summary is like, as I told, understanding the raw data is very crucial, and what are all the different NGS tools we know, we have to use them, understand them, and then we have to release a report. And NGS is not only used in oncology. I had given so much emphasis on oncology, but still it can be used in many other inherited diseases also. With this, I end my session. We can go on a questionnaire if anybody has any doubts.
Thanks a lot, Dr. Sneha. That was genuinely a very engaging and very insightful session. I
Thank
would
you, ma'am.
like to congratulate you for that.
Thank
And
you.
participants can unmute themselves and ask question if they have any. As far as I can understand, her lecture was very explanatory. So I guess there will be very less doubts, but yes. I request participants to ask any question if they have any.
Good afternoon, and thank you for the lecture. I would like to ask, I'm just getting to know about this next generation sequencing NGS towards late last year. I'm into malaria research, and I would like to know which of these two, like, will be the best, or which way will be the best to, let's say, analyze or run the genome sequencing for a malaria research. I'm presently working on the
Malaria.
malaria. Yeah,
Okay.
I'm presently
So,
working on mutating Anopheles mosquito with
mhm.
some selfish genes.
So you have already started or you're going to start, sir?
Um, I want to start this year.
You want to start doing NGS.
Yes, I want to start it this year. So I want to know how I can implement this into using
Sir, you already have any system or you're going to start fully fresh?
we are going to start fully fresh.
Then you can go for nanopore, sir. Oxford nanopore for infectious diseases. They are doing excellent job. And also it's a long read type of sequencing and time is very less. But I think it will be little expensive. I think so. I'm not very sure about the costing. But yes, it's a very long read you will get. And infectious, yes, they are doing a very great job. And your turnover time will be very less. Your bioinformatics help is too good. So Oxford nanopore is my choice. That comes under third generation. You can just Google Oxford nanopore. I think they have their if you're I think they have support system throughout the world.
Okay. Thank you.
Thank you. Yes, Shaurya, you can ask.
Shaurya, please unmute yourself and ask the question.
Hello, ma'am. It was a very great session. Ma'am,
Thank
can you
you.
please elaborate the use of machine learning algorithm in predicting the breast cancer or analyzing using the genomic data?
The software which I used.
Yeah.
Sure. So this is a commercially available software called Oncomind software. It comes with we are using in my company. I mean, currently in my diagnostic center, we are using Thermo Fisher. I'm sorry, I don't know what it. Sorry.
Okay, ma'am.
One second. Okay. Okay, I'm talking about this patient. What would you like me to explain, sir? What would you like me to explain?
I only want to know about the machine learning algorithm behind the use of these tools, bioinformatics tools.
No. See, this is a commercially available tool. So this machine learning, as I told you, you can use those Galaxy or any other tools where once VCF is come, then you have other variant interpretation tools also. In this, there is no
Okay.
machine learning. Like as such, there is machine learning, but I am not doing any machine learning.
Okay, it's on the back end. Yeah.
Sorry, sir.
It's on the machine learning is on the back end, right?
Yeah, it's on the back end.
Oh, thank you.
This is just an analysis software.
Hello, ma'am.
Yes, sir.
Can you just elaborate some points that why we need blood sample for germline mutations, not the tissue samples?
Great. So germline itself suggests, like say, especially in BRCA, okay, tissue is used to understand the somatic, okay. One thing is doing, efficiency or say simplicity is doing with blood, because whatever the DNA you're going to get, is of the same, right? If you're going to take DNA from a blood, it is going to be same. Okay. Whereas in somatic, when you're going to take tissue, okay, as I told, tissue heterogeneity is there. From there, it is going to be different. We have to understand that. Okay. That's why germline, you can go with a very less depth also, because your entire whatever DNA you have got is from the same source or same type of cells. Okay. So you can go to a very less depth also. Say I think some people do only 30x also. Say up to 100x it is advised for germline. Okay. And you will get very reliable data, because there's not going to be any, when it comes to tissue, it's not that only heterogeneity, there are other things also. If the tissue is not fixed properly, you will not get good quality DNA. Then there is something called as UDG, that is deamination of the tissue. So then in that cases, you will not get good quality DNA. Then you have to do, then sometimes there are situations where I've undergone that we would, ask the patient to do a rebiopsy. Okay, so germline variants is always advised to take from a blood, and doing germline from tissue doesn't make much of a sense, because tissue is mostly used for understanding the somatic nature of the patient, especially in breast cancer. In BRCA1, BRCA2, first thing they will do is germline testing, then they will go to somatic testing. Sir?
Thank you, ma'am. And also in your slides, you mentioned about this iron torrent, that it's generating BAM files, but most of the sequencers I saw, they generating FASTQ files. So do we have to convert BAM files to FASTQ?
No. FASTQ, from BAM you can convert into SAM, and then you can get your VCF file. Not required. FASTQ, no need to convert into FASTQ.
Okay, if I'm getting BAM files, so if I want to check the QC, so I have to convert them to FASTQ?
In case you're getting BAM file, say especially in iron reporter, if you're getting BAM file, you will get QC also. As I showed you those steps, right? There also you can do your QC. But in case you're using Illumina or say some other technique, then you will get a FASTQ file that you have to check the QC.
Thank you so much.
Hello, ma'am.
Yes, ma'am.
Are there any risk involved when we do the biopsy in the cancerous cells?
Ma'am, risk is there in every part of, diagnosis. But cancer, yes, you have to undergo a biopsy to tell if it is cancerous or not. That is the only true way of telling, yes, if it is cancerous cell or not. Okay, because sometimes you will also have benign tumors which may not be cancerous. That can only be told by biopsy. Yes, you have to do a biopsy. Risk is definitely involved, but not very, very much. But yes, risk is there, but we have to do it. And the procedure is not easy at all. If it is going to be lung, it's a very difficult case. Even if it's going to be breast, yes, it is difficult for the patient to take up the procedure, but that is how it is.
Okay, ma'am. I would like to ask one more thing. Hello.
Yes, Priya? Yes, Nashrin?
Ma'am, actually, it's all about my grandmother.
Okay.
Once she was very much ill, so we got to know that she is suffering from cancer. But after that, from 2 years, she was much more healthy than before. Like no any symptoms of cancer was showing, only just at the time of when she was more ill, and now again she is again ill. So like what is happening? That
So
there is
what
many
type of
signs and
cancer was it?
symptoms. It
Which
was of
part?
like thyroid part.
Thyroid. Okay, maybe it is metastasized now. So
Okay.
you definitely have to do a thyroid biopsy. Was the biopsy done earlier?
Yes, ma'am, it was done earlier.
Okay, then you will have to take her for a PET CT.
What is that?
PET CT is a type of CT scan. So there it's a procedure where they will know if the cancer is spread elsewhere or not.
Okay.
If it is spread to lymph nodes, it's going to be difficult. Her age?
Like 80.
Hmm?
80 years.
80? Then you should just leave it. That's it. That's that, you know, any more treatment procedure is going to, you know, it's going to make her more ill at this point.
Okay, ma'am. Actually, she is taking homeopathy medicine.
Okay.
Yes, ma'am.
There is no comments on that.
We understand that this is somewhat a very emotional thing for you, but it is what it is. She is 80 plus, and perhaps all of the diagnostics and all of the medicine will not work on her. And I guess doctor can suggest best of the solutions. And as Dr. Sineha has guided you that you can go for PET scan. Any other question? Yeah.
No, ma'am. Thank you.
Thank you. Ma'am, there are some questions in the chat box as well. Please address them. And I request everyone to mark their attendance because we will not be able to provide certificate if the attendance is not marked in the sheet.
Article, Mohammed Sashin has asked for some article about male breast cancer. I can definitely share. I will definitely share to Deepshika ma'am. I think you can share it to him, ma'am. That will be doable.
Yes, ma'am, definitely we will share.
Okay. Sai Adira, what are the bioinformatic needs in the cancer? How, with the help of bioinformatics, can we diagnose the cancer? This entire session was that only, I think.
Manzar Hussain has raised his hand. Please unmute yourself and ask the question. Manzar.
Hi, madam. I have a
Hello, sir.
question. What are the key difference between inherited genetic mutations and acquired mutations in cancer?
Sir, inherited is something, you know, that you have got it from, say, your parents, or say from, it can also be sometimes that it has been there from the first cell of you, okay, when you were even an embryo, okay. Acquired is something that you've got in the due course of your life. And that's what we call as somatic. Somatic is because it will not be there in all of your cells. It will be there only in particular few cells in your body, only in few, you know, that distorted cells. Whereas a germline or an inherited variant will be there in all of your cells. And to detect that, blood sample is enough. For detecting your somatic, you need tissue.
Then, what kind of medicine we use for inherited and acquired mutations? They are the same or different?
Sorry?
Madam, what kind of medicine we should be used for inherited? Same kind of medicine we can use for
Same kind of medicine.
Medicine. I
Good,
thought
good,
you
good.
not a medicine.
Very nice question. So germline, they do. Yes, of course. Mostly the chemotherapy would not change. But especially in breast cancer BRCA testing, when they do a germline testing, we can also say if it is a generational effect. Okay. By that, what I mean is, say if a woman, okay, is affected, is carrying a BRCA1 variant, we always advise, once, if we know that she has a, you know, if she has children, we advise them also to get a germline testing done, which will help them that, you know, anything we cannot say, you know, say, oh, you will never get cancer. No, Okay, in case it is being detected in the children, we can never say, oh, you won't get it. But we can prolong it. Okay, so in case if the child will get it at 40 years, maybe we can with the lifestyle. And there are some chemotherapies that, I'm sorry, not chemotherapies, chemo drugs that can be advised for them, that it will prolong the occurrence of the cancer. So that's why generally when a germline BRCA comes positive, we immediately counsel the patient to get their children. Say basically if they are above 20, it's more good to get a BRCA testing done.
Thank you, madam.
Anything else, sir?
Sir, clear. Madam, clear.
Mr. Muhammad, please unmute yourself.
Good afternoon.
Good afternoon, sir.
Yes, so you've talked about the types of variants and it can be inherited or we can get it in our life. So as the type of variants do we so can we have both the variant both in both cases? So is there a variant can be inherited and also can be developed in
Yes,
in our
highly
life?
possible. Yes, highly possible.
Yes, thanks.
You are not audible, Muhammad. Mr. Hamdan, please unmute yourself.
Hello, first, thank you so much.
Yes.
I would like to ask, you mentioned before that the germline it's better to take the germline from the blood cells than the rather than the tissues. True for both cases, whether the cancer is inherited or acquired in the two cases.
Sorry, I didn't get your question. Sorry.
Okay, so you mentioned that the cancer could be inherited or acquired. Yes?
That is a variant. Okay. The variant can be either acquired or inherited. Yeah.
Okay. In both cases, the germline should be taken from the blood cells, not the tissue.
In what cases?
The someone before me asked about the germline. Germline, if it should be taken from the blood cells or tissue, and you mentioned that it's better to take to do the germline test from the blood cells rather
Yes.
than the tissue.
Yes.
Okay, so my question was whether this radiant was inherited or acquired. In both cases, that germline test we do it with the blood cells, not tissue cell or tissue.
In case it is inherited, it is advisable to do using blood. As I told you, doing with the blood will give you better results. Okay, better results in the sense better data. I shouldn't say the word results, better data. So it is advisable to take a blood sample. Okay, in case your blood comes negative, then we'll go for tissue. And in tissue, say in case if the blood has no variance, then if a variant comes in tissue, we cannot say it is, mostly we can't say it is inherited.
Okay, can I ask you why we could have better data or data from blood cells?
Because you're going to take that DNA from WBCs, or say the DNA source is from a single type of intact cell. in the case of blood. Whereas in the tissue, it's not the case. Okay. In the tissue, we have lot of issues. As I already mentioned, when they are fixing the tissue in FFPE, there can be some issues. Okay. Which may lead to a very bad quality tissue. From that, when you extract a DNA, it is again going to be bad quality. Then next comes tumor heterogeneity, which I already told. Not all cells are going to be of the same character, right? So the differences in the gene will happen. DNA will happen. Okay. That time what happens? The mutation shown by one may not be shown by other. Then my allele will come down. Frequency will come down. If my frequency is going to come down, then how sure am I going to report that variant as? So in that cases, blood is always, analysis wise also, blood I can confidently very easily tell. So for inherited, it is not advice. Generally, they take blood only. And if you want to know if it is somatic, then you have to take tissue only.
Okay. Thank you so much.
Thank you.
In the interest of time, probably we would not be able to take more questions. Thanks a lot, Dr. Sneha.
Thank you,
I
ma'am.
request all of you to please send your questions in the attendance sheet, because this is also a feedback form. And we will send that to Dr. Sneha. Probably she'll find time to answer them. Okay. So people are asking what other services we are providing except these monthly webinars. So I would like to share my screen and will let you know about what Dr. Omics is and what we people are doing. And Dr. Sneha, please let me share my screen.
So I should stop sharing, right? Yeah.
Obviously, yes. This is
Sorry,
the
ma'am. Thank you.
Thank you, ma'am. Thanks a lot. Yes, we will share recording and PPT with all of you. We will mail you. Okay. Just a second.
Hello.
Yes, we can hear you.
Ma'am, your
Yes.
attendance sheet is not open.
Yes, I know that. Thanks for intimation. I suppose that you can see my slides now. Just a second. Is it visible?
Yes, ma'am.
Yes, ma'am.
Yes.
Thanks a lot. Okay, so we are a private limited company registered at Delhi. Our name is Dr. Omics Labs. This name will be pronounced not as Dromics. We are Dr. Omics. So we are a molecular diagnostics service lab, and we are specializing in molecular diagnostics and genomics. The basic motto of our company is that we provide genetic predisposition of various disease risk along with drug resistance, heart health, overall wellness. And recently we have launched a very exciting genomics-based test, that is hair genomics. So we would like to contribute to pharmaceutical research and other type of bioinformatics trainings as well. Basically, there are two verticals of my company. The first one is diagnostic lab, and the second one is research lab. During my journey, I felt that there is a huge gap between academia and the industry. We are educated. Yes, we are. But still, after getting very good education, we are not able to get the required job because there is difference. What this industry is expecting from us and what academia is teaching us. So we decide to bridge this gap, and we come up with this Dr. Omics Research Lab vertical. Here we provide training and education along with research and development. To be very precise, in this vertical, we provide bioinformatics-based research and trainings only. We don't provide any other type of internships except bioinformatics-based internships. Because generally people are also asking for microbiology and biotechnology-based internships. No, we are providing dry lab-based internships only. So we are incubated at prestigious IIT Delhi. We are recognized by Startup India. We are ISO certified, MSc certified, and recently we have received $5,000 grant from Amazon Web Services. We have successfully trained over 5,000 students till now. And yes, we are a bit popular on LinkedIn. Apart from that, the most important part for our vertical 2 is that we have signed a 5-year-long MOU with Andhra Pradesh government. Andhra Pradesh is a province in India for those who are foreigners. So we are committed to provide internship for more than 10,000 students of Andhra Pradesh. In the Dr. Omics Molecular Diagnostic Lab, we have different type of diagnostic tests available. These all are saliva-based tests. We will require only saliva from your side. We will extract the DNA, and based on different type of SNPs present in your DNA, we will identify cardiovascular diseases. We can diagnose cancer up to some level, and type 2 diabetes, and 10 major drug resistance and overall health guide is also present. Recently, we have introduced 2 different type of long-term industrial bioinformatics internships. Both of these internships are 6-month long. On 22nd of January, our recent NGS-based internship will be launched. In that 4-month intensive coursework will 4-month intensive coursework will be there, in which we will teach you from very basic. We will start from how to fetch data from different type of databases. We will teach you Linux. Apart from that, there will be classes of Python and R. And after that, rather than Galaxy, we will teach you command line based pipelines. And it will cover 4 different type of data analysis pipelines, including DNA-seq analysis, RNA-seq analysis, both de novo and reference-based. Along with that, there will be microarray data analysis and metagenomics. After that, there will be project work, 2-month-long research-oriented project work. And for first 5, the topper of the group, we will guide them to publish a paper. We will not only guide them, we will guarantee that their project will convert into a research publication. For rest of them, we will guide how to publish the paper, but for first 5 toppers, we will guarantee that their project convert into a publication. So I have already told about this. This is our team, Dr. Sudarshana. She is scientist and PhD in bioinformatics. Need not to say that every person has done their education from bioinformatics background only. We are bioinformaticians, short and short. So Dr. Risha, Dr. Om Prakash, you people have met him in last webinar. He is very learned fellow. Dr. Swapnaja, Dr. Jyoti, Sushma, Dhanshri, you have also met her during our previous webinars. Bhushan is also Python trainer. He is very dynamic. Priyanka, Akshay, Samriddhi. So yes, this is Dr. Omics, and I welcome you all so that you can come and see what type of internships we are providing, what type of test we are providing. Please go and visit our website. And if you are a bit curious that how we are going to teach you, then first 2 classes we are providing free of cost. During your attendance, please mark whether you want to receive the Zoom link for the same. If you say no, we will not send you the first free 2 classes, but